Hello everyone, and thank you for joining us for the College of Engineering Faculty Lecture Series. The 2024 College Engineering Faculty Lecture Series features lectures by faculty members leading groundbreaking research in the areas of wastewater recycling, desalination, mineral extraction, water quality management, and more. Today's lecture is titled Revealing Nature's Secrets, a chemical forensics approach to decode the information stored in water. The lecture will be presented by Gerrard Jones, Assistant Professor in the Department of Biological and Ecological Engineering. My name is Leslie Rojas, and I am a student in the College of Engineering, and I will be today's event moderator. We encourage you to submit questions throughout the lecture using the Q&A button at the bottom of the Zoom screen. A recording of the lecture with captions will be provided to all audience members after the event. If you need assistance at any point during today's event, please put it in the private chat to the host and moderators. With that, it is now my pleasure to introduce Adam Ward, Department Head of Biological and Ecological Engineering.
I'm pleased to introduce today's faculty speaker, Dr. Gerrard Jones. Gerrard's interests lie in maintaining environmental health through multiple perspectives, including a wildlife biologist, environmental engineer, and environmental chemist. Throughout his career, Gerrard has focused on how environmental phenomena are governed by interactions between physical, chemical, and biological processes. His research focuses on water quality, using machine learning algorithms to understand the links between landscape processes and the resultant water quality impairment. He's also interested in soil geochemistry and understanding how human health is directly linked to environmental processes. These processes and their control on trace element concentrations in soils is of particular interest. Gerrard, thanks for joining us today. I'll turn things over to you.
Hi all. Thank you very much for the introduction. I'm happy to be here and to share some of my research with you. But a little bit of my background. I'm a Quechan tribal member, and the Quechan people have a rich history of exploring the natural world and studying it in great depth, the world that we live in. And so I've added to this with a variety of degrees in biology, wildlife science, environmental engineering, and inorganic geochemistry for my postdoc. And so these are all seemingly unrelated fields, but the single common theme that has always guided my research is what environmental factors drive, and insert your favorite environmental phenomena here. So these different aspects of my background just really give me different tools to understand the world that we live in. And so my master's degree, I was studying wildlife biology, and the nice thing about wildlife biologists is that they use, and ecologists, is that they use multivariate statistics to help understand the systems that they work with. And so this has been something that has stuck with me over the years, this reliance on multivariate statistics to understand what's going on in the environment. For my PhD, I was looking at the fate and transport of endocrine disrupting compounds from agricultural fields. And here I was used a lot of physical modeling tools to understand how chemicals move through the environment. And for my postdoc, I was looking at some machine learning tools to understand trace element concentrations in soils. And this was really, these machine learning tools were really useful for linking the biological, chemical, and physical processes that drive these phenomena. Now that I'm at OSU, I do a lot of environmental chemistry. And so here we study the chemical universe. Physicists, I've always been a little envious of physicists because they have these real charismatic tools like the electromagnetic spectrum. Biologists have the tree of life, but you never hear of similar aspects of their study for environmental chemists. And so as an environmental chemist, I study the chemical universe, which is I think of it as a romanticized term to really describe the vast array of compounds and elements that exist throughout the cosmos. And so I really like this image because it looks like space. It looks very dynamic, but really this is a gasoline drip on pavement. And so it's a really nice lead in this connection of this chemical universe to gasoline and pollution in the environment that is really at the center of my research. And so really the main theme of my research is understanding why the chemicals are present, understanding the pattern in the chemicals that are present in an environmental sample. And so in this paper that I found during my postdoc, there are 20,000 unknown chemicals that are present in a single wastewater sample. And when I look at this, I don't see a random pattern, or I don't think of a random pattern. Really what comes to my mind is what story is hidden in this data? What can we learn by understanding the chemicals that are present? And so my overall hypothesis is that all biological, chemical, and physical processes have distinct chemical signatures. And so a little bit of background, there's been a tremendous amount of work focusing on problematic chemicals, ones that are known to cause environmental harm. And so recently, scientists from Washington discovered that chemicals in entire particles are lethal to coho salmon within a matter of hours. And this work took over a decade to really identify this one chemical. And so if you think of all the chemicals out there, there's a lot of time investment that needs to go into these type of these analyses to really understand the threats that chemicals have to our environment. And so from my perspective, even if we know what the chemical, the problematic chemical is, or even if we don't know it, we still need to do source tracking. From my perspective, once we identify a source, then we can implement some type of engineered practice to put in the environment in order to remediate that pollution. So instead of finding one in tens of thousands of chemicals, maybe we can find the one in potentially dozens of sources. And once we know the source, we can implement an engineering strategy to solve it. And so for some of the most problematic pollution sources, like, or the most problematic and prominent pollutants, such as nitrate, identifying the source of nitrate is more important than identifying that nitrate is problematic, since it has many sources. And so there are likely some organic molecules that are diagnostic of each source. And once we know a source, we can prioritize these water management solutions. And so my overall hypothesis is that given the diversity of organic molecules, the probability that any two sources or environmental processes could produce identical chemical signatures is virtually zero. And the prediction is there exists some combination of chemical features that are diagnostic of every process and source. And so this has really been the focus of my research for the past several years. And so today I want to share 3 projects with you. One is, can we develop a diagnostic chemical fingerprint for point sources of pollution? This ended up being relatively easy. Can we develop a chemical fingerprint for a landscape? This ended up being pretty hard. And can we divide the overall chemical composition into meaningful subunits? And this is some ongoing work that we're doing. And so the ultimate goal for pollutant source tracking is to collect a water sample from the environment, run it on some instrument, apply some machine learning classification tool to be able to figure out or predict whether or not some pollutant source is present and whether or not others are absent. And so this was the overall workflow of one of my early master's students, and I'll walk you through each of these steps here. And so for the sample collection of this project, we focused on 4 or 5 pollutant sources that are known to contribute to nitrate pollution. And so we had agricultural runoff, street water runoff, animal manure that we collected from various locations throughout Corvallis. We went to the city of Philomath, where we collected water from their wastewater treatment plant, which was a series of ponds. And then as a background sample, we went to high elevation locations in the Cascades as well as the Coast Range to collect some creek and lake water samples with little urban development within the watersheds. Next was running it on the instrument. So we took the water samples into the lab, extracted the organic molecules, and loaded them onto a high-resolution mass spec in a shared instrument facility here on campus, and processed the data. Ultimately, what we had from our 50-ish samples was about 7,500 non-target chemical features. And a non-target chemical feature is anything and everything that will hit the instrument detector. So here we had about 7,500, and these are distinguishable from each other just based on their retention time in the instrument, as well as their mass to charge ratio. So we used a variety of machine learning tools to distinguish or to try to identify which chemical features best distinguish one source from another. And so what I'll talk to you about today is using support vector classification. This tool ended up being real easy, is real fast to run this. An advantage also of support vector classification is it gives us a coefficient weight for each chemical feature. And these weights can be positive or negative, and they're really indicative of the predictive power or the ability that chemical can predict the presence or absence of our sources. And so here's a plot showing the ranked coefficient weight. And so these weights can range from -1 to +1. If you have a chemical feature that has a coefficient weight of +1 or is more positive, it's typically more predictive of presence. If it's closer to -1, it's more predictive of absence. And if it's close to 0, there's very little predictive power of that coefficient weight. And so if you want to develop a diagnostic chemical fingerprint, really you want to select the chemical features that are present in these tails. And if you're around 0, you're really not diagnostic of that source. And so for our 5 different sources, we arbitrarily picked the top 10 chemical features that were in these tails here. And using just these top 10 chemical features, we're able to predict presence or absence of all of our samples with greater than 97% cross-validation accuracy. And so this was really exciting. But we probably shouldn't be too surprised by this. If you think about it, we expect wastewater to be different than street water runoff, to be different from ag runoff, to be different from manure. And looking at this plot over here, we have our 5 different sources, and this plot summarizes the overall chemical composition of our samples, and we see that each of the sample types cluster together. So it's not too surprising that we can develop a diagnostic chemical fingerprint. The real utility of this technique and approach comes when we want to screen an environmental sample for the presence or absence of one of these sources. So that's exactly what we did. We sampled 4 different creeks in the area, Oak Creek and Dixon Creek and Mary's River in and around Corvallis. And farther north in the city of Dallas, we sampled Rickrell Creek. Each creek had 5 sample locations, moving from upstream to the left, downstream to the right. And we collected samples at 3 different seasons, spring, summer, and winter. And we tried to collect our water samples at different transitions between the forested headwater areas, the agricultural areas, and some of the urban areas. And so we had some expectation that we might be able to detect each of our chemical fingerprints within these samples. And so, are the chemical fingerprints detectable in the environment? In this plot, we have our 5 different source types, our 3 different seasons, and then on this y-axis, the probability of presence. And the most interesting data was from Rickrell Creek. And what should jump out at you is this relatively high probability of presence for these blue bars, which is wastewater treatment plant, specifically at locations 4 and 5 in the spring, 4 and 5 in the summer, and maybe even 4 and 5 in the winter. And so we didn't know it at the time, but when we looked at a map in between locations 3 and 4, we thought there might be a wastewater treatment plant. Lo and behold, when we look for it, we see there's the Dallas wastewater treatment plant. So we were really excited. This was the first proof demonstrating that this technique works and is useful for detecting the presence or absence of a source in the environment. So what does this all mean? The chemicals in the environment are not random. They carry information about the sources that are contributing to the chemical composition. But I think it also points to this idea that water bodies are data loggers. You can imagine there's chemicals washing downstream every second, and they carry information about what's going on upstream. And so it's up to us to go collect that information and extract that useful information and translate it into useful knowledge based on the chemicals that are present. And so with a single water sample, we could potentially quantify all upstream sources. And so this tool could be useful for watershed managers. Once developed, a watershed manager can screen for any and all fingerprints at the same time for just tens of dollars per sample. And so it could be a real efficient way for watershed managers to start monitoring water quality in watersheds in our communities. I also want to point out that it's easy to make assumptions, particularly with nitrate. The agricultural community is often blamed for nitrate pollution. Certainly there are sources of nitrate coming from agriculture. But I want to point out that our ag runoff, the probability of presence was fairly low throughout all of our samples. And so this was I want to highlight that this approach can be used to exonerate folks who are often blamed for contributing to pollution. And it's important for us to check our assumptions because they're not always correct. So this work is published a couple of years ago from my first master's student, Manuel Davila Santiago. So moving on to the next question, which is to fingerprint landscapes. Certainly landscapes do contribute to the chemical composition in surface waters. And so we're able to fingerprint a source, but can we fingerprint a landscape? And so I just got through highlighting that we can fingerprint different sources, but that's only useful if we know ahead of time that that source might be present. In this example over here to the right, these are creeks that were those creek samples that were influenced by wastewater. And when we look, these creek samples plot with all the other creek samples. So overall, the water still looks like creek sample. It doesn't look like a wastewater sample. And so it highlights that the wastewater isn't the most prominent source contributing to the overall chemical composition. There's some other source that is making all the creeks look like creek samples. And maybe that's landscapes. And so the question is, how do you sample a landscape? There's no single point in time or space that is forest or sagebrush steppe. And so landscapes represent a continuum over space and time. And so it really makes sampling challenging in these locations because you can't go to a single spot to get a representative sample. So instead we used our creek samples, and the idea was there's some gradient in land use change. All these creeks start in these headwater areas and they transition through landscapes, they transition through residential and urbanized areas. And so we thought maybe we can identify changes in the chemical composition that correspond to changes in the landscape. And so our hypothesis was that overland runoff contributes to the chemical composition in surface waters, particularly in the winter rainy season when overland runoff carries chemicals to surface water. So we made a variety of predictions that we expected. Sites with similar land use, so forest sites would cluster together, ag sites would cluster together. Summer sites will cluster together because there is no runoff from the landscape, so it might just look like a water sample instead of, say, an overland runoff sample. Landscapes will be most different in winter due to the runoff, and shifts in the chemical composition will correspond to major changes in land use. So we had a very similar chemical processing. In addition to the chemical data, we used some satellite data and stream stats to get a wide variety of environmental variables such as temperature, precipitation, as well as land cover variables. In addition to our chemical data, we also had we collected some microbial samples to see if the microbial community composition could be contributing to changes in the chemical composition. And so, as a reminder of our predictions, here are our 4 creeks. Each creek has a different land cover composition. This green is forest, this yellow is agricultural, and red is urban. And so we expected all these 4 sites to cluster together, the urban sites to cluster together, and et cetera. And we expected major chemical shifts as you traverse the landscape, as you went through different land cover types. But we saw none of this. None of these predictions were seen. Instead, we saw 3 major clusters by season. And within a single season, each creek clustered together. Each creek was more similar to itself compared to other creeks, regardless of their land cover. And so it's certainly not land cover. Season seems to be important, and maybe even this might point to unique ecosystem processes that are occurring in these different seasons. And so we see this very similar clustering when we look at the microbial communities. Instead of 3 distinct clusters, we see 2 distinct clusters, a winter cluster and a fall and spring cluster. And so when we look at the microbial community, here in this plot, we show the percent of the microbial community and where that microbial community is coming from. This blue area this blue color is associated with freshwater communities, and so phytoplankton. So in the summer, there's a lot of the freshwater microbial community present, and in the winter, there's a lot more of this gray community, which is a soil microbial community, and so that might be water leaking into the rivers from nearby soils, bringing those soil microbial communities into the water column. So we did a structural equation model, which is a fancy linear regression. And I don't want to go into this too much detail, but really I want to call your attention to these numbers. The greater the absolute value of these numbers, the greater the importance it is. And so the largest value are from our environmental variables. So things like temperature, precipitation, some of the seasonality has the largest effect on the chemical composition. A close second is the microbial community. And probably a distant third is the effect of land cover on these chemical composition. So environmental variables are important. Environmental variables also influence the microbial community, and land cover was relatively weak. And so what does it all mean? Ecosystem processes may control the overall chemical composition more than the discrete sources. My assumption is that land use was an important driver of the chemical composition is likely incorrect. This doesn't mean that landscapes are unimportant. They're just not the most important contributor to the overall chemical composition. And so how do we fingerprint a landscape or a process then? So far, the supervised machine learning and our ordination tools were unable to identify the chemicals derived from these sources. And so really what it means is we need new computational tools to identify which sources have the greatest contribution to receiving bodies of water. So this work was published by a recent graduate, a recent PhD student, Chang Shi. Okay, so far we've learned that the overall chemical composition is affected by several sources and processes. And we know that there's a strong seasonal variability. And so this makes sense. As the seasons change, seasonal processes change, and so we expect that the chemical composition to vary in time and space. And just because one process is important at one time and space doesn't mean that it's going to be important in the next. And so really I'm trying to lead into our next question, which is how do we divide the overall chemical composition into meaningful subunits? A subunit that might be important in the summer, a subunit that might be important in the winter, or two subunits that sum to equal the whole. And so far I've expected to find preconceived patterns. I've expected that chemical sources would be different. I expected that landscape would be different, or landscapes would be important. And so this points to this idea of I have knowledge about the system, when really I should go in thinking that there's information out there that I don't know. So I should let the data speak for itself before I impose my own biases on what may or may not be present. And so we need a data-driven approach that helps us divide the chemical composition into its most important subunits. And to do this, we use non-negative matrix factorization. And so this is useful for dividing the data into the most meaningful subunits without imposing my idea of what may or may not be important. So we have a collaborator at Cornell, and so we use some previously published data. Briefly, this data is collected at the Fall Creek Monitoring Station in Ithaca, New York. And it collects water from this Fall Creek watershed, which is largely agricultural. And so we collected weekly samples, or the samples were composited on a weekly basis for about a year and a half, from March of 2017 to September 2018. In this data set, we had just over 3,000 non-target chemical features that we were able to detect. And similar to the previous study, we used satellite imagery to collect a wide variety of watershed characteristics in order to help us explain the chemical composition in these weekly samples from this monitoring station. So looking at the overall summary information present in the samples, what we see here, similar to what we saw before, is that there's some different seasons. These dots are color-coded based on the season that they're collected, and these larger dots indicate the average condition for each season. And so we see that there's kind of this shift in the chemical composition. I was expecting that I was expecting kind of a circular pattern where the springs would be more similar and the summers would be more similar, but we see kind of this continuing shift, this continual change in the overall chemical composition, which might indicate that processes occurring in streams might be continually changing, which has some significance for climate change. As environments change due to climate change, the processes in the stream might just continue to evolve and change through time, which is interesting but separate question. So looking at this data, we see that our flow rate in our system is important, as well as the temperature, explaining the chemical composition in our streams. But really, I want to spend a little more time looking at the most important subunits. So using this NMF technique, this non-negative matrix factorization, we identified 5 subunits, and the idea is that each subunit is indicative of a different process or source that contributes to the overall chemical composition. And so you can see that each process is important at different times in the year. There is no one single process that dominates. And so this was unexpected and surprising that there are several processes and sources that have a major influence just at different times of the year. So looking at our 22 environmental variables, we did a correlation with each of our NMF variables, And there's really only one variable, NMF2, that is strongly correlated with any of these variables. And so NMF2 seems to be related, strongly correlated with some snow variables, snow melt, as well as negatively correlated with things like water temperature, soil temperature. And so this seems to be the most seasonally influenced variable. So we looked at some of the correlations, jumping to NMF1. The most challenging part of this NMF analysis is then to interpret what each of these factors mean. And so some of these were more easily to interpret than others. NMF1 ended up being a fairly easy one to interpret. Plotting NMF1 as well as the wastewater discharge, we see a reasonable correlation with the peaks in both peaks, or sorry, the peaks in both of these plots. We also discovered a wide variety of chemicals that were present, things like pesticides or pharmaceuticals that were present in our NMF1 data. And so the presence of these chemicals suggests that this is potentially wastewater discharge. So pesticides end up in our wastewater treatment plant when things wash into the gutters, and then they go downstream to the wastewater treatment plant. Pharmaceuticals coming from our wastewater treatment plant are often untreated, and so they end up moving through the wastewater. And so we've got some decent evidence to indicate that NMF1 is related to wastewater. It gets a little tedious to go through all of these NMF factors, and so I'll just give you the highlights. As mentioned before, NMF2 is likely related to snow melt or some type of hydrologic process. NMF3 is likely agricultural because it correlates with a variety of agricultural pesticides. NMF4 was correlated with water temperature, pH, and alkalinity. So this one is a little tough to interpret, but it might be related to groundwater processes, geochemistry. And NMF5 is likely related to natural processes because it was correlated with soil temperature, evapotranspiration, and organic carbon, this UV 254 absorbance. And so not all of these would be something that I would readily predict or say, I bet this hydrology variable is really important. And so interpreting these NMF variables ended up being really insightful into adding to our understanding of what's driving the chemical composition in this system. So what does this all mean? We can decompose the overall chemical composition into the most important subunits instead of assuming what they are. This ended up being the most challenging part of the work that I've done at OSU over the last 7 years. And so it's still in its early stages. We're still just scratching the surface into how we can take this chemical composition and interpret it. And we really need new techniques. So working with computer scientists and machine learning and statisticians is really important for us to be able to improve water quality. So even though it's challenging, we're starting to make some headway to understand what the dominant what processes are driving the dominant chemical composition in surface waters, which is a big achievement. So even though it's challenging, I don't want to diminish our accomplishments, specifically the accomplishments of my students. So my overall conclusions, the goal was to extract useful information from large chemical data sets and transform it into knowledge about chemical sources about the watershed. This is useful because we can quantify the presence or absence of different pollution sources. And if some source is disproportionately high, say wastewater or ag runoff, this allows watershed managers to direct limited resources towards projects that might have the biggest impact on water quality, the biggest improvements to water quality. And I will say that this area, it's a research area called chemical forensics. This is largely unexplored because environmental chemists, environmental engineers, ecological engineers don't typically use multivariate statistical and machine learning tools. And so there's a big need for folks to study these fields because right now it's the Wild West. And there are a lot of examples of poorly applied statistical analyses which can lead to erroneous or poor management decisions. So it's really important for water quality engineers to have this expertise. So overall conclusions, I never thought that I would need machine learning, multivariate statistics, mass spectrometry, GIS, microbial ecology, computer. There are a lot of things that I never imagined that I would need to study. And so I just want to highlight that there's a big need to continually learn new skills. Once you're done with school, it's not over. And so there's always this need to collaborate with people and think of new innovative ideas and how the tools developed in another discipline can be used in your discipline. So if I could start all over, I might just do data science instead of the trajectory that I took. But the only reason why I got here is because I was curious. I explored things that I was passionate about, and I seized opportunities to collaborate with people and learn new skills when they came along. I've got a large lab that I want to thank, particularly the three people at the top, Manuel Davila Santiago, Gauri Mahadwar, and Chang Shi, who are the primary engines and drivers of this research, as well as a large host of supporting students, as well as faculty at OSU, and funding that has helped me get to this spot. So thank you all very much. Appreciate it. And with that, I'd be happy to answer any questions.
Thank you, Professor Jones, for sharing your research with us. I learned a ton, specifically with the chemical composition, and I think what stuck with me was the surface water bodies are data loggers. I think that is a quote that I'm going to be thinking about every time I go into a river or even to a body of water or sea. But we will now begin the Q&A portion of our event. Remember, if you'd like to pose a question for Professor Jones, please submit it using the Q&A button at the bottom of the screen. We also have some advanced questions submitted by the audience. We won't be able to address every question today, but we'll get to as many as we can. We'll begin with a question I wrote down during your lecture. The first one is there a specific story that stands out the most from the chemical composition of a sample that you've been able to do?
Stands out the most to me personally, or I think the biggest story is what I try to emphasize. I had this expectation of what I would see, and I didn't see that at all. And it was the influence of microbes. And so there's 2 points I want to make. One is just my own bias. I went into it thinking I would find something, and my student was like, no, that's not correct. And so I was glad I was open enough to realize that it was a really important discovery that really challenged the way that I thought about the ecosystem and stuff that happens. The other thing that I want to point out is, of course, microbes. When I was an undergraduate, that was the part of my biology background that I liked the least. I was like, I will never study microbiology. I'll never use this. And it turns out to be one of the most important aspects of this story. And so it highlights really the importance that microbes play in shaping our environment. And when I tell students that microbes are important, sometimes they're like, oh, because they're not the most exciting, or at least visually they're not the most exciting. But in terms of their implications for water quality and the environment, they're definitely very, very important.
I think all of this research and all of what you're saying is, it goes to show how much you're willing to also learn despite your background. Like you keep learning, like you were saying at the end. And it doesn't end in school. It just keeps going after.
Oh yeah, absolutely. Especially with this, chemical forensics, it requires me to be able to talk to chemists, talk to microbiologists, talk to engineers, talk to health folks. And so, there's always this need to learn more so I can communicate this information and refine my approach to just learn more about, how our environment works.
All right. We have a question, and it's what a low-cost ways can neighbors band together and test local soils when the local government says it's not possible or that there is no need for testing or verification?
Yeah, that's a great question. I have there are a wide variety of ways to answer this. And one example is to reach out to OSU Extension. And so the Extension Office has a variety of tools for helping people test all kinds of stuff from nitrate or lead in their water to asbestos or soil, chemical contaminants in soils. And so I think I've seen soil testing, maybe not advertised, but on the Extension webpage. And so reach out to the Extension Office, and I would point that person to reach out to them, and they'd be able to help find a specific test that they might be looking for.
Thank you. Our next question is the nitrate runoff from cows similar to what would come from other wildlife?
That's a great question. And so if you look at Benton County, one of the top reasons why streams in Benton County are in violation with the Clean Water Act is because of bacterial pollution. And so E. coli in specific, I know this is related to nitrate, but the two are related because they come from animal waste. And so Benton County streams, if there's going to be a violation, it's probably going to either be temperature or bacterial pollution. And so I tried to create a chemical fingerprint for different sources of manure. So we had, I think, cows, horses, pigs, ducks and geese, chickens, turkeys, and then wildlife, ducks and geese. And so we're trying to make a chemical fingerprint to distinguish the different sources of manure. And so we collected those samples, ran them on the instrument, and we have yet to analyze the data. So I would expect, based on diet, that the chemical composition of these animals are different, but I can't tell you with any certainty at the moment. That's my expectation, but we know how my expectations turn out.
Okay, we'll look into it, I guess. Our next question is, will computing facilities like the HCIC help interpret subunits?
What is the HCIC? I presume that is a high performance fancy computing cluster. If that's true, then absolutely. Specifically, the expertise needed to program and run these analyses. We're getting increasingly larger and larger amounts of data, and my laptop can only handle so much before it's just computationally infeasible to do these analyses. And so I think collaborating with professionals and computing clusters who can, not only computing clusters to help us run the analyses, but also specialists in data analysis and machine learning to leverage those computational tools. That's just something I've never had any real exposure to, and I'm very limited. And so I absolutely welcome those collaborations from folks who can all things computer science, computing. Welcome collaborations from folks in those spheres to help out, because there's obviously a lot to do. And with my background in environmental science, it's a barrier that I face all the time. So definitely it'll help out.
Thank you. It looks like we have time for one more question. Do you see or have you seen applicability in determining effectiveness in vegetative buffer or other mitigations from roadway runoff pollutants?
That's another great question. And the answer is both yes and I don't know. When I was doing my PhD, I was looking at, one of the very first slides I showed this trend where there's kind of this downward trend through time. And so vegetative buffers on the edges of ag fields, the edge of roadways, they are very useful for removing contaminants. Just the organic carbon, the vegetation creates a real sticky environment, and chemicals will stick to that organic carbon and are effectively removed or prevented from entering the stream. So definitely a very useful tool for treating specific contaminants. What I'd really like to do is run a chemical fingerprint through there to see how much of the overall chemical composition changes. And so it's a very active field of research in my lab right now to look at the change in the chemical composition as a measure of treatment. And so I've never run like a manure water sample or a stormwater runoff sample through a vegetative filter strip to see how much of the chemical composition changes. So we know that certain stuff absolutely is removed, but I can't tell you how much of the overall chemical composition changes as it runs through a vegetative filter strip. So very, interesting question, and one that definitely warrants, more investigation.
Okay, thank you. Yeah, one more question. The 7,500 data points showed linear patterns. Is there a chance that these line of dots are really indicating a common single constituent or a family of compounds that just vary in terms of how the compound got partially consumed?
Absolutely. And so I always thought of them as tiger stripes. When you look at that plot, you see just these kind of threads of chemicals. I can't tell you about all of them, but some of them are absolutely repeating units, where you just have one, carbon, hydrogen, or carbon oxygen group that just repeats. And so it ends up being very related chemical compounds that just have a slight retention time and mass shift. So you get these, they just kind of walk across the screen as the molecule continues to expand. So a lot of those are, they're called homologous series, where you get very similar chemicals that behave very similarly in the instrument. And so you get this very predictable pattern in the output of your plot.
Okay, thank you. And another question is asking, can this information be used to raise awareness without raising alarm? That is, what can we do to engage a proactive public rather than a reactive public? If that is outside the scope of this presentation, I understand.
Yeah, that's another excellent question. And it's always a goal of mine to engage with members of the community, industries. There is a lot of concern. Nobody likes it when somebody's like, oh, I'm an environmental chemist, I want to test your water quality. Nobody is really excited to have their water tested, especially if there's this history of people pointing the finger to say, oh yeah, ag is really, really problematic. X industry is really problematic. And so I really want to engage with these industries, particularly agriculture. I rely on food. I like eating food just as much as the next person, and I'm not out to shut any one farm down or impose restrictions on any one industry, just because we're also dependent on these industries. Really, my goal is to engage with people. I get excited about water quality problems simply because the engineer in me is like, okay, what type of simple, cheap solution can we implement that a farmer, rancher can manage themselves, that will improve water quality, that will be good for everybody, and also maybe help them get ahead of any type of regulations, so that they're better equipped to deal with regulations that are coming in the future. And so absolutely, there's a huge need to engage with the public. A good place for that is working with extension people. Also means that I need to also be a good steward and go out and talk to people and listen to what their concerns are, listen to what their needs are, and bring those concerns with me back into the lab, so that, yeah, I don't just close people off and exclude them from the conversation, but also give them a voice, so that science can be done in the best interest of the community.
Okay, we have another question. Given that there are hundreds to millions of chemicals present in most surface water samples, what are the most sustainable and regenerative, included affordable ways to prepare surface waters to recharge of groundwater aquifers?
That is a really great question, and I don't know the answer. And I don't think anybody knows the answer. I mentioned earlier that this was an active area of research looking at the overall change in the chemical composition as a measure of treatment. And so we're just now starting to do some experiments that try to address this very issue. You can imagine that something like reverse osmosis, which is probably the best available technology that we have for cleaning water, is also a very expensive and impractical technology. So there's this trade-off of what's going to be readily implementable and what's going to be effective. And at this point, I don't think anybody knows the answer. Nobody's done those experiments to say, okay, here's what the vegetative filter strip can do. This is what the infiltration trench can do in terms of changing the overall chemical composition to remove contaminants. So very, very, we're at the very beginning stages, and it's something that definitely needs to be done. And I'm hoping to see, especially as students leave the lab, as people see their papers, I hope they start looking at us and like, oh, how can we test this this treatment train to look at the change in chemical composition? That just hasn't happened yet because this research is so new.
Okay, we have time for one final question. Where do you see the techniques being most feasible to include in regulations?
Another great question. So I had a conversation with shortly before this work was published, I was at a Southern Willamette Guama groundwater management area, a Guama meeting, and there were two regulators, and one was like, oh, this is going to be so powerful for regulation, and the other was like, this is going to be a nightmare for regulation. And so this idea of regulation is really important. And so in the back of our mind, yeah, thousands of chemicals. Right now, this approach can only be used, not only, is most readily applicable with high resolution mass spectrometry instruments, which aren't readily available at state agencies. And so it's still an area where research labs and universities that have access to these tools can do this non-target chemical fingerprinting. So it's really up to us to translate these tools and develop them using instruments that are readily available by state agencies. So we're still in the research phase. Next steps would be to develop them using more widely available instruments. And at that point, then once we get them down to an instrument that can be used at a state lab, then it can be implemented into regulations. And so still a ways to go, definitely in the back of my mind. And so we're still in the research and development phase. If I was to guess at a time frame, maybe 10 years out before it's written in any type of regulation that a chemical fingerprint can be used by a regulator. That's just a really back of the envelope guess at the earliest, but I'm aware of it and moving towards that direction that this could be used by a regulator in a fair and equitable way.
Thank you again, Professor Jones, for the fascinating lecture and for your answers to all the questions. Before we go, do you have any parting comments to share with the audience?
Other than thank you very much for your questions, the one thing I'd like to say is stay curious, and continue to learn about computational tools, machine learning, AI tools. They're really useful, and I'm hopeful and optimistic that the next generation of grad students come through the lab are like, oh yeah, as a hobby, I've done a lot of this work, machine learning. I've built my own computer games, and I want to apply it in environmental ecological engineering applications. So maybe that plug is for younger audience members. But yeah, reach out to me with questions. I'd be happy to answer them offline as best I can.
Thank you to all of you in the audience again for joining us today and for your great questions. As a reminder, we will email you a captioned video recording of the lecture. Thank you again for attending and have a wonderful afternoon.